Okay, good. Welcome, everybody. It is my pleasure today to welcome Dr. Pouya Bashivan today. He is a recent arrival at McGill University. He started in 2020, and he's one of the QLS supervisors in the PhD program in Quantitative Life Sciences, and we're really happy to have him. He did his postdoctoral fellowship, some of it at Mila and some of it at MIT. He has a PhD in computer engineering from the University of Memphis in the USA, and is working on models to predict brain responses. And we are looking forward to his presentation today. Thank you so much, Pouya. Please go ahead.
Thank you very much for the introduction, and thanks for inviting me here. I'm really glad to be here and telling you guys about what we've been doing. So most of the topics that I'm going to discuss today, they're coming from like the two postdocs that I've been working, but the stuff from my own group still have not been coming out. Hopefully next time around, I'll be talking about what we're doing, what my students are doing. All right, so so today the topic is about artificial neural networks and visual neuroscience, and I wanted to tell you guys about some of the work at the intersection of computational modeling with ANNs and how we could apply, or we've been applying them to neuroscience to try to explain what the neurons do, mostly in the context of visual object recognition. All right, to get started, what do we mean by visual object recognition? So if we try to define visual object recognition as a behavior, then visual object recognition is a task of naming objects by just looking at images, right? And as humans, we're extremely good at doing this kind of task. We're able to name among many hundreds or even thousands of objects. We're just like briefly looking at images, which are really very high dimensional inputs for a visual system. And the object recognition is considered as a very challenging task, but maybe as a first thought, it might look as a trivial task that could be solved by a method like template matching. Like for every object, we could have a template, and we just like look for the template, whichever template matches best is that would be the object. But when you look deep down, it's a vastly complex task. When you start considering all the different variations that every object could have, and to start thinking about like the fact that every object can create infinite number of possible projections onto the retina, which is essentially the pattern that our brain sees, right? And the challenge of object recognition is essentially to know or learn how to map all of these different input patterns to the same label. In this case, like all of these variations of the car that you're seeing, a 3D model of the car, regardless of what size it is, like where in our visual field it falls, and like whether we're looking at it from the front or from behind, like whether it's rotated or not. I try to have a system that calls every one of these images the same thing, a car, and not a dog, for example. And this paper from 2008 by Pinto, Cox, and DeCarlo gracefully demonstrates how the amount of variation actually is very important in how challenging the task of object recognition becomes. And this paper was originally kind of like a critique to the data sets that were common at the time, namely Caltech 101, if I'm not mistaken, and criticizing that like having like a very small amount of variation is not going to be useful for finding a solution, a brain-like solution to object recognition. Because just if you imagine all the objects, for example, have the same view, like side view, if you look at the top side of the figure on the right, then the task of categorizing these images into two categories of like cars versus planes would be rather trivial. So at a level that even like a very simple model, like the V1 model, would be almost perfect at solving this task, even if we just randomize the background to figure out how to recognize it. But now, as you increase the variations allowed in these kind of like two-dimensional projections of these 3D objects, it becomes more and more difficult for a simple model like V1 model to solve this task, to a degree that when you have a lot of variations, like in this case, the classifier is essentially reduced to like 50% of random chance. Now, to motivate you a little bit more about why object recognition is important, or what I'm calling invariant object recognition is important, let's think about a hypothetical scenario. Let's say we have an animal, a mouse, and this mouse lives in a very simple environment. And like most animals, our mouse lives to survive and lives to reproduce, which is a little bit contradictory, self-contradictory, but still nevertheless mostly true for most animals. And let's say that in this environment that our mouse lives, there are two types of objects, two classes of objects. One is the class of cats, which are the predators, and the class of cheese, which is the food. And the mouse has to learn that in order to survive, it has to find cheese and go and eat it. And then whenever it sees a predator like a cat, it has to run away, right? And the whole process of learning this, learning to survive, involves knowing that every pattern that's the instances of the cat and cheese might create on its retina, on the mouse's retina, it has to be learned that all of those patterns have to be corresponded with some kind of policy, some kind of action, like this particular instance of a cat, you have to run away, but also this instance, and also this instance. And these are just examples, but as I said in the previous slide, there are an infinite number of possible projections of every object onto the retina. So is the case for the cheese, like whether it's small or large, it doesn't have as many holes itself, right? And if you imagine the direness of the environment that the animal is living in, it becomes very costly to start the animal to start learning all of these projections one by one. And it is very likely that the animal at some point will die even of hunger, or just not being able to run away at the right time from the predator, right? So this would be the scenario under which the animal does not have any kind of object recognition or invariant object recognition system. This is what the animal has to do. Now, if we move to another scenario, which we're assuming that the mouse, this animal, can invariantly recognize objects. So what is the benefit of doing it? So if the animal is able to do this, then it can invariably recognize that all of these patterns correspond to the same label. And all of these, for example, cat, and all of these other patterns, they correspond to the label cheese. And now all the animal has to learn is to learn the simple policy that if I see a cat, then I have to run away. And if I see cheese, I have to run towards it and go and eat it, right? But that's not everything that comes out of this kind of system, this kind of architecture and learning. Once you have this kind of representation, then you can ideally generalize too, meaning that now if you see a relatively different pattern of a cat, a new type or breed of the cat, then it's more likely that you can quickly understand this is also another predator that I have to run away. And whether the cheese is white or yellow, it's still a cheese and it would be tasty and have to go and eat it to survive. And also going further, the animals can start to build a knowledge base around those objects, meaning that now it can quickly learn how a cat sounds like. So even without seeing a cat, it would be able to recognize that there's a cat around and try to run away as soon as it can. Also, how a cheese smells like, or how does a cat move? And these are just examples. Like essentially, you can build all kinds of representations on top of objects. Feel free to ask questions, interrupt me if you have them. Now, going to the neuroscience of visual object recognition. In the primate brain, object recognition behavior is facilitated by this pathway called ventral visual pathway, which starts from the back of the eye, from the retina, and then ends at an area called inferior temporal cortex, or IT. And in the middle, going from the retina to the tip of IT, there are several cortical and subcortical areas that are involved in like essentially producing this kind of representations of objects that are invariant to all of those transformations that we saw in earlier slides, the ones that we called identity preserving transformations, right? And now to understand how this visual pathway is enabling us to do this behavior, neuroscientists have been looking at what the neurons do in each of these areas and try to find clues of like what's happening from the retina to IT that is enabling us to do that. And a traditional approach has been to look at what individual neurons in each area are most selected for, meaning that's like what kind of visual patterns they respond to. And from the literature, from groundbreaking work from Hugo and Wiesel back in the 60s, we know that the neurons in V1, which is kind of like back of the brain, like one of the earliest stages of visual processing, we know that the neurons in this area, they respond best to light bars and dark backgrounds. And different neurons, they respond to different orientation of bars, like some of them to vertical bars, some of them to horizontal, and different degrees and so on. And then we start moving forward towards the tip, and we get to area V2, and by the time that we get to area V2, it's kind of like already getting a little bit complicated, complex to describe what V2 neurons are doing, what kind of patterns they're responding to. And the best description for neurons in area V2 is kind of like a texture-like pattern that are generated by correlating the output of V1 type of neurons. Like if you imagine V1 neurons, they responded to different orientations. Now you combine different orientation, different patterns, you get textures. And we start moving even further, we get to area V4. And in this area, the pattern is continuing the trend of becoming more and more complex. Now in this area, the neurons, they get more selective for very particular instance of curvatures or complex gratings, if you will. You can look at some of the examples of like what I mean by curvatures or gratings in this figure. It's like there are all kinds of like or patterns, like so that you can name, or some of them are difficult to name. But the important fact is that they're now becoming like multi-parts as we move forward. And this pattern becomes more and more evident when we start looking at IT neurons, because by the time that we get to the tip of IT, you start to see neurons that are highly selective for multi-part objects like patterns, like the ones that you see in this image. And another interesting fact is that from the posterior to anterior, this pattern still continues, meaning that in the posterior IT, selectivity patterns are relatively simpler compared to anterior IT. Now, if you want to summarize everything we saw over like a quick overview of some of the literature related to neuronal selectivity along the ventral visual pathway, you see that as we're moving away from the eye, from the retina, the type of patterns that these neurons are selected for, they're becoming more and more complex, right? But at the same time, it's becoming kind of more and more complex to actually say what these neurons are selected for, because the possible space of patterns are becoming larger and larger. If you compare V1, which was essentially the space was different orientations of light bulbs. By the time you get to the space that the IT cortex is representing, the object space is so large that it's like becomes exponentially difficult to find the instances of the best objects each neuron is selected for. And now, another note that I wanted to mention here was that, like, you look at the neuroscience literature, a lot of the kind of explanation that is being given in the literature is kind of like a verbal type of explanations, right? And the verbal explanations are very desired because they're easy to communicate. You can easily talk to everybody about like what it means for a V1 neuron to do something that it's doing, right? But at the same time, we have to appreciate that the verbal explanations, they have narrow bandwidth. And what do I mean by that? For example, if you consider V4 neurons, you can easily tell someone that the V4 neurons, what they do is this respond to curvatures. And just that short sentence gives a very good high-level sense of like what's happening in V4, right? But now if you want to expand on it, if you want to tell about like what happens in the population of neurons, how does each neuron work, and like how does neurons transfer information between each other with spikes, and talk about channels and so on, then now you have to fill books with all of that information, right? And it might take like weeks or months or years to really understand like what's happening and what do V4 neurons do, right? And this requires something called literacy. So you have to be literate in order to understand with detail like what's happening and what do V4 neurons are doing, right? And maybe one more downside of verbal explanation is that now there are always more questions to ask, and their answers are not really immediately clear from the literature. But maybe you can say that you can find experts that have spent years doing research, and you can ask those questions from those people and get some answers without doing new experiments, right? But maybe a lot of them, even considering those situations, a lot of questions may still need experimental, costly experiments. Now, in the contrast, there could be also quantitative explanations, right? Here, I'm mostly thinking about neural networks, but it's not a statement about neural networks in general. It could be any quantitative model that we could use to predict responses, for example, or explain what the neurons are doing in terms of their activity, right? So what I want to argue here is that these quantitative explanations, like those in neural networks, can also be communicated easily. But at the same time, they're also easily transferable. And what I mean by that is that if I want to tell you what a V4 neuron is doing, or a V4 equal of a V4 neuron in a neural network is doing, I can probably give you a short sentence like the one that I have on the slide. V4 neurons in my model are formed by training a CNN to classify objects using backpropagation, right? Like the short statement that we had from the verbal explanation doesn't give you all the information, but gives you a relatively good high-level understanding of like what's happening, and so on, right? And now again, comparable to what I told you in the verbal explanation side, now if I want to really give you all the details of like what operations are involved, how are the parameters updated, what data set did I use, and so on, like what objective. Now I said they're easily transferable. And what I mean by that is that now I can give you the code. I can give you the data set, and then you can go and look at the code and see what's happening inside the code, and even try to run the code and see the results on your own. And this is what I'm calling like now, in order to understand what exactly the computational model is doing, we need to have some level of coding literacy, right? Just kind of like in contrast to the classic literacy of like being able to read and write and so on, right? But maybe there would be some pushback in this point, but I thought there would be an interesting kind of discussion if somebody's interested. And now another point about the value of these models is that now if you have a model that is really good model of these neurons, you can go and query them to answer new questions without actually doing any costly experiments, which is, in my opinion, one of the best benefits of these kind of neural network models of today. And now, because I wanted to mostly talk about artificial neural networks, just kind of give you an overview of like where things are coming from, like where did they get started and so on. So ANNs are not new; they've been around for a long time, and like one of the starting points of where they were introduced was this work by McGill and Pitts that they came up with the first artificial neuron, aiming at kind of explaining what an individual neuron might be doing from the computational point of view. And then later on, that work was extended by Rosenblatt into a multi-layer perceptron, which is essentially like putting all of those single neuron models together into different layers of computation and getting into a multi-layer computing system, our first artificial neuron. And then roughly from that time around, of course, I'm removing a lot of the literature in the middle, but trying to like create some kind of flow. it went mostly two ways. One was for the neural networks for vision, which was kind of followed by models like HMAX for the visual perception of the brain, and then more recently by deep neural networks. And then the other way was for memory and decision making with mostly recurrent neural networks. Again, worked with Remo Hart et al. And then more recently, Monte and Cecilo's work on using recurrent networks to kind of explain what neurons in the prefrontal cortex do in decision making tasks. And but not much was surprisingly looked at until maybe the last decade or so, that people started to really directly compare the activity of the units in artificial neural networks and the activity of neurons in the brain. And then trying to create like one-to-one mapping between these artificial units and real neurons in the brain. And the gist of findings was that these I'm calling them DNNs here, deep neural networks, that people found that the representations learned in the layers of these deep neural networks are highly predictive of the activity of neurons in the brain. And the results that I'm showing here is mostly from two papers. Fortunately, I forgot them too. Yeah, I have the citations here, but I forgot to put them here, coming from these two papers, looking at monkeys and humans, putting everything into a short sentence, like you see in this slide that the activity of these neural networks are able to very well predict the responses of neurons. The red curve is actually predictions. The black curve is the actual neuron firing rate to different images. And you see that there are many different images, natural images with random backgrounds. And another fact that you can see very quickly is that the deeper layers in the neural network, they correspond to later stages of processing in the visual cortex. And in this figure, you can see the two areas, the V4 area and IT area and macaques. You see that the best area that represents the neuronal responses in V4 is like between area 2 and 3, but IT, which is anatomically is downstream from V4 area, is best corresponding to a later layer in this neural network, which had only 4 layers. And the same story roughly in humans. I'm going to show you some results from humans from our work also. But yeah, overall, there are a lot of similarities between these neural networks and what seems to be happening, what kind of representations the neurons along the ventral stream represent. Now, despite all of this, there has been one kind of common and well-known criticism of the kind of representational similarity analyses. One that has to do with the correlative measures, right? So everything I showed you from those papers, what they do is like they correlate the responses between the unit activities and artificial neural networks and the neural activity in the brain, right? And you could say that because these DNNs are very high dimensional, even despite using cross-validation strategies, there might still be chances that only because of the mere high dimensionality of these networks, that some of these correlations are spurious and actually are not what the neurons represent, right? So the question becomes now, how can we go beyond the correlative measures of similarity, right? And this takes me to the first paper that I want to discuss today. So one direction that we thought of going was to kind of see if these kind of models can enable us to exert control over the system that we are studying, right? And the idea of control comes from model-based control theory, right? In which we usually have a system with its own inputs and outputs, and we have a controller, or we want to have a controller that, given a desired output that we imagine for the system's output, controller could give us some control signal that if we let the control signal input to the system, it will give us the desired output, right? And now the idea would be to imagine the neurons in the brain as the system that we have a model of when we want to exert control over, and to think about the controller as our ANN, the predictive model of the brain. And now the question we want to ask is, can we use predictive models like the ANNs that we discussed so far to control the neural response patterns in the brain? And how are we going to do this in practice? We're going to use the model to generate new visual stimuli that drive the neural population response into desired states. Now tell me, like, let me tell you like a gist of like how we're going to do this. Like, what is this image synthesis procedure is about? So roughly there are 4 stages involved. The first stage is that we take a neural network model with a particular architecture, and we go and tune its parameters so that it's enabled to do an object recognition task on a relatively large scale data set. Once we're done doing that, then we fix all the parameters, and then we set on a row to create a mapping between the units in our artificial neural network and the neurons that respond, right? And the way we do this is generally by taking a data set of natural images and then show those images to both our animal and to our neural network model. And we get the activation from different layers of the processing or ANN. And at the same time, we'll record the neural activity in an area that we're interested in the animal brain, right? Now, having 2 data sets, one of the neural activity, another of the artificial neurons units activity, we can create a mapping using simple methods like regression. Essentially, mapping the activation in a layer of a neural network to the brain responses. Once we do that, now we have a full model that for every image, given pixel values of every image, it can make a prediction of like what those neurons would respond. What is the neural response level? And now the third stage is we use an optimization method to use the model that we have, which is a differentiable model. I'm not going to go into the details, but like roughly what we do is that we use our model and follow the gradient so that when we have an objective function that gives us the desired pattern of output or the neural activity, it can generate some input that would predictably give us that desired response. And the fourth stage is having those images, we go back to the animal, we show the animal those images, and we validate how successful it has been. Oops. I'm sorry. All right. So what I want to tell you today is 2 scenarios that we've tested this idea. One scenario we're calling stretch or maximum drive. In this scenario, we're considering, if you consider the responses of one neuron to different images, natural images, as a distribution of this blue distribution. What we wanted to do was to use the neural network model to try to find stimulus patterns that would drive the neural population response beyond this normal, the regular population of natural images. And that's why we're calling it stretch, because we were thinking of it as like stretching responses beyond the natural stimulus. And first of all, let me show you, like when we did this kind of mapping between an artificial neural network and the neuronal responses in area V4 in this case, what did we get? So in this plot, scatter plot, I'm showing you the predicted and measured neural firing rates from one V4 neuron to a set of naturalistic images. And you see that the model is doing a really good job at predicting how that neuron would be responding to these naturalistic images. And these images have been evaluated using cross-validation. So we're not really using the train set. We're not using the predictions of the train set. And now the question was that we wanted to see whether we can use this model to now push the responses of this neuron beyond this distribution. Now, the distribution, if you imagine, would be like projections of all of these dots into the y-axis, right? And if we now go and look at like what the best natural image for this particular neuron looks like, we see something like this, like an image of a plane. And here I'm zooming in into the receptive field of this particular neuron, essentially the part of the visual field that this neuron has access to. You see that this is essentially the pattern that this neuron is seeing. Let's it's really difficult to explain what this pattern has. It might be like a set of concentric circles here and an angle here and so on, right? And now when we used our methodology, when we synthesized and we controlled the images and we showed it back to the animal, we saw that indeed these images were able to push the neuronal firing rate beyond the distribution of naturalistic images. And now we were curious to know, what did these images contain? And when we started to look closely, we saw that, again, we saw some patterns that we could relate back to what we were seeing from actual images. Like you could think about these patterns as a set of concentric circles, but it's not the whole story. Also, like different corners, like the ones that you see here with a particular angle. But as a whole, the combination of all of these patterns gave us the higher response in this particular neuron. And you see 5 synthetic images here. The synthetic images, the difference between them was that when we were synthesizing them, we started from a different random seed. But all of these 5 images, they all produced high responses in our target neuron. Although, if you look at the pixel level, they're highly different from each other. But perceptually, you might say that they have a lot of similarity to each other too. And what we wanted to quantify here was to see, like, how much have we been able to really push the neuronal firing rate beyond what we could see from our natural image set. And this was our indicator of like a benefit of the degree of benefit that you could expect to gain from having a neural network. And for this, we just computed the firing rate from our best natural images and the difference between our best synthetic, and kind of like looked at the ratio. And overall, we saw that we could stretch the responses in the before neurons by about 40% when we looked at an average of around 16 minutes. Now, the second scenario was even more ambitious. So what we wanted to do here was, unlike the first scenario, which we were only focusing on one neuron, we're trying to do a similar version of this, but on a population of responses. And in this case, if you imagine the neuronal responses in a population of 3 neurons, they look like these dots in 3-dimensional space and as a ball, what we wanted to do was target one neuron, like N1 here, and try to push the responses in N1 while also controlling the responses of N2 and 3 so that they would not really deviate from the natural distribution. So keeping the responses in N2 and N3 really small, close to the natural distribution, pushing the responses in N1, our target neuron, as high as we could. So this scenario, we call it one hot population, or OHP, which we'll see in the slides. And as you can imagine, now you can go and change the target neuron to any of these neurons that we have at our arsenal. So before I show you like how the synthetic images look like, the responses, let me show you what the response to our best natural image was. So this image was selected so that it would kind of like give us the closest sense of the desired response we had in mind, which was essentially this red the red line, which is like maximal activity in the target neuron. It's like smallest activity in non-target neuron. You see that the best natural image is actually not doing a great job, meaning that it has a relatively high response in the target neuron, but it has a lot of responses in non-target neurons too, right? And again, I'm showing you here an extended receptive field of this neuron just to get a sense of like what this neuron is seeing within this receptive field. And now, when we synthesize the synthetic image and we show it back to the monkey, this is the kind of response that we got from this procedure, which I'm sure you will appreciate is like much closer to what we wanted to have, a desired response here, right? And again, to measure how successful we were, we defined this loss term, objective term, which is essentially the same loss function that we use in our optimization for image synthesis too, which is a softmax function. Try to maximize the response in our targets and minimize the response elsewhere. And one final point is that although I showed you 2 scenarios with 2 particular objective functions, but this is by no means only limited to these 2 objectives. You could think about any objective. You could think about these objectives as questions you want to ask, and then you want to use the model to kind of answer those questions. And another related experiment that we did was what happens if we consider only a population of neurons with highly overlapping receptive fields, and we repeat the procedure. And again, we see that we're highly successful at individually controlling each of those neurons when we target them separately. And final point here is that we also observe that as our models are becoming more and more accurate, the level of control, you see here that it's a scatter plot. On the x-axis, we see a measure of functional fidelity, which is essentially how predictive is the model of the neuronal responses, and each dot is 1 neuron. And on the y-axis, we see a measure of control success. We see that the more accurate models we have, they enable us to do more accurate control. It's just kind of already maybe given, but this is a confirmation. And interestingly, at around the same time, 2 other papers came out looking at essentially the same concept in area V1 and IT. So there's a type of it has to be IT. And they kind of looked at neural network models, trying to find the best kind of patterns of stimulus for neurons in these 2 areas. And they found very interesting patterns, like mice. They found that it's not only about orientation selectivity, but the patterns are like mixtures of many different things that are also equally difficult to explain. Also, the same story in IT. They found these like really weird images that seem to be like the best drivers of IT. Now, to recap what we've been talking about so far, we started by asking, can predictive models of brain enable precise control of neural responses? And I showed you some evidence that we can, at least in area V4, we can get a decent level of control. We can exert a decent level of control over the neural population response using this type of neural network models. So, any questions up until now? No questions? All right, proceed.
You can put them in the chat if you want to.
Yeah,
I'll keep an eye on it.
Yeah. So where do we go next? One direction that we started exploring was, now everything we talked about so far was done in macaques, physiological data, which are relatively they have high SNR. Now we wanted to see whether the same kind of approach could work in principle in humans using data that is much noisier, fMRI data. And we know that much of the human brain is visually driven. So if we show like a bunch of images like we did with the macaques, and you should look at the boxes in the brain and how which ones are really responding to natural images. We see that a lot of the brain, especially the ventral side of it, is lights up. But it's not only ventral; it's like dorsal also, but maybe more responsive to motion than static images. But overall, there are a lot of the cortical areas are visually responsive. And there's also a fair amount of category selectivity across the cortex we know about from the literature. One of the most well-known category selective regions in the human brain is called FFA, or fusiform face area, which originally it was found in a study by Nancy Canwisher et al. By looking at the contrast of the fMRI bold signal when subjects were looking at images of faces versus images of non-faces, like O here, or objects. And they found that there are these actually several parts in human subjects that they respond selectively much higher to images of faces compared to images of objects. And essentially the statement here is a distributional statement, meaning that there are these voxels or population of neurons that if you look at their responses to all images of faces, overall they respond higher compared to other categories like places, bodies, or in general objects. And again, this is a summary of what it looks like. If you just look at the mean of these responses and you try to put them in a bar graph, then this is the kind of pattern that you get for a face selective area. And the selectivity is defined as a distributional difference in brain responses to different categories, right? But this finding was not really only about FFA areas. Later studies show that there are also other areas like area PPA that are highly selective for images of places or scenes. And also the body selected areas that seem to be responding much higher to body parts like arms, fingers, and legs, and torsos, and so on. But within all of these studies, no matter how many times they've been replicated, these results are always attained by using statistical significant tests and relatively small sample sets, right? Maybe a handful of images, tens, maybe not maximum of hundreds of images, right? And this leaves the claims open. That could there be any untested stimuli that would refute the prior category selective accounts of these areas? And this is only possible if we have the capability of testing many thousands of millions of images, right? Ideally. Now the question is, how can we test these theories against millions of possible images? Because like we know that with modalities like fMRI, it's very difficult and time-consuming, costly to get responses to all of those images. And this is the work that was led by Ventral in collaboration with Alex Bates, Jim DePaul, and Nancy Campish. So the gist of it is that we again used the same computational approach that I told you about earlier, similar to other work from Dan Yames and Nicholas Krivoskortis' lab, which is essentially like considering a set of natural images. We show human subjects those natural images, we collect the data, the fMRI data, while they're reviewing those images. And at the same time, we can show the same type of images to our neural network model. In this case, it's a different architecture, it's around 50, and then create a mapping between the unit activity in the neural network and the activities in different parts of the brain, cortical. So we built these models similar to what I already explained. And then next, we went ahead to check their predictions, right? So the first place that we looked at was the phase selective area, the FFA in human subjects. And we found out that the similar procedure can again lead to very accurate models of the responses in this area. And the accuracy was not necessarily only the difference between face selective and like faces and non-faces. And you can see here, color coding the images of faces and non-faces, and in showing all of these predictions along with these actual measurements for 4 different subjects. You can see that the predictions, even within different categories, like category of faces and category of non-faces, including like places, body parts, and objects, they're kind of equally predictive. They have very high correlation, meaning that they could essentially capture the variations within different object categories themselves too. And it's not only limited to cross-category predictions of like whether this is a face or not. And the same story was true in other 2 areas that we looked at, namely EBA and PPA in both hemispheres. And this was done only using the same model, ResNet 50, but from mappings to different areas according to like which region we wanted to map onto. Essentially, we picked the best layer of computation. And if we now pull everything together, we see that the first column here is showing us the noise ceiling in the data, which is the cap at which we can hope our model to really predict those responses. And we see that if you consider all the stimuli when we're predicting, we're very close to the noise ceiling, meaning that there's like not much left to be predicted or explained within this data set of around 200 images. But if we focus our analysis on within different categories, then we see that the gap is really larger. So it means that essentially predicting all the variations across different instances of every category is more difficult. And this is just like an extrapolation of the data that we've seen here. Now, going back to the original question we had, we wanted to see whether, like, we were looking for a way, essentially, to find the answer to whether there are any images that would refute the theories of selectivity, category selectivity in these areas. So for that, we used our model to kind of do a version of this experiment without actually collecting the data. So what we did was that we brought in the model and we let millions of images from three standard benchmarks in machine learning and computer vision that include a lot of images of faces, places, and in general objects. And then for every one of those images, overall, it was about like several million images that we ran through the model, we looked at the predictions for each of those areas. And then what we did next was we looked at all of those predictions, and naturally we see that for an area like FFA that we know is selected for faces, the distribution of predicted activations is much higher for the data set that contains mostly faces, PGD face. And for body area, it was mostly the same. And for places, we also saw that the data set contained mostly places and scenes, the green curve, produced the highest responses. But this was not really enough. We wanted to look at like individual images and make sure that we don't really have any images that would refute those theories. And what we did was we also looked at the top driving images within each R1. And for that, we went over several thousands of the top images that were predicted to have the highest response within those areas. And we looked at those images for each of those three areas, both hemispheres. And we found that when we even went down to like 25 and 100,000 images, we still saw that the best images for each of these areas were essentially the theorized images for those areas, like faces for FFA, and mostly body areas, including fingers, torsos, and legs and arms for EBA, and for PPA, mostly scenes and different places. But even despite screening through millions of images, one might say that there might still be some image that was not in those data sets that we haven't tested. So to kind of like push the nail even further, another experiment we did was try to do a version of what we did in CACs to try to synthesize images for each of those areas and see what kind of images would appear out of a synthesis procedure that we really don't have any biases towards what should be the category of objects. And again, we found out that the images coming out of a synthesis procedure could easily be identified with the category that was originally thought about for each of those areas: faces for FFA, body parts for EBA, and places for PPA. So now to summarize, computational models evidently confirm prior accounts of computation in various regions along the ventral stream. But do these models add anything to what we already knew from these theories? So to kind of push this a little bit further, we wanted to ask two questions. Do computational models provide a more accurate account of ROI function beyond classical theories? Classical theories, if you remember, were about the category selectivity. This area is responding to faces, not responding to others, right? We wanted to see whether the model is actually going beyond this or not in any sense. And the second question was, do computational models provide a more accurate account of the ROI function beyond what the human expert's knowledge already indicates, right? To go after the answer to these questions, we did a set of behavioral experiments. But we had two cohorts of subjects. The first cohort, which we're calling the M Turkers, or Amazon mechanical Turkers, we collected their behavioral responses to an experiment which they were asked to rate how category-like are these images. For example, showing images of different faces and non-faces and asking them to rate how face-like they are. And a similar experiment was done on human experts, that had published extensively on one or all of those three areas. And then, for example, we asked them a more precise question of predict what the FFA response would be to each of those images. So they had to rank order the images with respect to the predictive responses that each of them would generate in FFA. right? So we considered the subject pool 1 as the responses from the subject pool as kind of like indicator of category selectivity responses, and the subject 2 as like indicator of what the human expert essentially indicates in terms of like accuracy of predictions. And we did this just to give us a way to compare these kind of predictions with the model predictions that could be made on individual images and predicting the responses in each ROI. And so again, I'm putting these 3 groups here, calling them novices, experts, and A and N. And overall, we found out that when we looked at all the stimuli within a pool, within a data set, the 3 categories were not that far from each other for FFA and for EBA, but a little bit more for PPA. Especially the novices were much less accurate at making those predictions. But the story became a little bit different when we started looking at individual categories, going within the stimuli for faces, bodies, objects, and scenes. And we found out now, if you look inside each of these categories and then you consider only the variations within the stimuli within each category, like all instances of faces, all instances of scenes, and so on, now the model is mostly much more predictive of these, accurately predictive of these responses, even compared to human experts, which were good overall over all images, and mostly because a lot of the variance comes from the differences across different categories. For example, here, the differences between body or non-body body images. But then if I showed all the images of the face, of faces, they would not be as good in predicting like how would EBA respond to those face images. And we concluded by saying that seemingly the evidence shows that the computational models provide a more precise account of the function of all ROIs compared to classical theories as well as the expert knowledge. And in the interest of time, just one final slide I'm going to go over was another experiment that we did, trying to inquire from our model to see that if I'm showing a model one image, what would be the parts of the image that are real drivers of the responses in any of those areas that we were looking at. And for this, we used a technique called RISE, that essentially given an image, it creates many masked versions of this image, like the ones here, and then makes a prediction of the response for each of those masked, half-masked or randomly masked versions of the image, and then combines them together to get this heat map of the importance of different parts of the image in making the prediction go high. For example, making the prediction of FFA to be high or low. And then we applied it on a set of different images that to get insight of like, where exactly is this model getting its activation from? And then surprisingly, or maybe not so surprisingly, we found that indeed the model was looking at, or if I may use the word attend, was attending to what we thought was the relevant part of the image for each of those cortical areas. For example, in this case, you see that most of the activation, high activation area FFA was coming from the part of the image that was corresponding to face and facial features. Also here and also here, which is pretty interesting because in this image, person is wearing a mask, and also only the part of the face that is not wearing a mask is effectively driving the FFA response. And also for the other areas like EBA, you see we have hands here, torso here, and also like mostly neck and paw here. And for PPA, mostly like around the background, the scene, and everything that is around. Which is kind of like confirming that the model is essentially looking at the feature that we thought, even from the beginning, should be driving the responses in those areas. And I have more slides today, but I don't think I have the time to go over all of them. So I'm going to stop here with this summary of the slides that I discussed so far. So feel free to ask questions.
Thank you so much, Pouya. There is a question in the chat from Curtis. I don't know, Curtis, do you want to turn on your microphone?
Yeah, yeah. Can you hear me?
Yeah.
Yeah. Okay, so the earlier part of a fantastic talk, Pouya. I really, really enjoyed your talk.
Thank you.
Very good. The first part of the talk about the stretching the neurons responses was very, very striking. So you can design a stimulus that drives an individual neuron much better than the great majority of natural stimuli.
Yes.
I was wondering, are there interesting behavioral responses from the animal? I mean, what you're doing there reminds me of the ethologist concept of a supernormal stimulus. People like Konrad Lorenz and Nico Tinbergen in the 1950s showed many examples of this. They didn't have an algorithm. They just came up, if you give an extra large egg to a bird, will it preferentially sit on that and neglect its own eggs? And the answer was yes, they will.
I see. To be honest, the behavior that the monkey was doing was no behavior. It was just like sitting down and then looking at these static images. So they were not asked to do any kind of behavior. Later on, before I leave Jim's lab, we did some limited experiments on trying to control the monkey's behavior by changing, for example, the balance and arousal of images, and then training monkeys to like do a version of that task. We had some success with that, but the farther we went away from the neurons that we were targeting, it became more and more difficult to actually control that variable, the latent variable that we were thinking we were controlling. Right. Which is of course understandable, because there's still like a lot of circuits, neuronal circuits between behavior and what we are recording from in V4 and IT, right? So it becomes a much more difficult problem to solve. And just another note, even going from V4 to IT using the same models, the predictions and these measures of control become less successful too, because mostly we think that our models of IT neurons are really less accurate than V4.
Right. I can see it's problematic designing a behavioral experiment. A simple idea would be just simply to look for signs of, did these stimuli give greater arousal to the animal? So like if you're doing eye tracking, most eye trackers track pupil, you could look for a small pupil dilation. It's, wow, that was interesting. That was a super face,
Right. And at the same time, like in that experiment that we were doing, sometimes the model was doing trivial things, just like saturating colors and so on, which was kind of like not that interesting if that was all the model was doing. So at some point it became very tricky to say like whether we have anything interesting here or not. But I agree. I think the most complex part of it is to design an experiment that would be really meaningful in a way that can really show or highlight the power that you can gain from having a model like this.
Right.
Thank you. Other questions for Pouya? You didn't talk too much about how difficult it is to get the data that you need for this, although you did mention sample size near the beginning. So what's involved in that process?
Yeah, I had some size on those, but in the interest of time, I just got rid of those. I thought like it would be better to talk about some conceptual ideas. But yeah, so the monkey experiments were kind of like done in a closed loop system, as you can imagine. So the way it was done was that we would collect data to natural stimuli on one day. Then we would quickly go back, process the data, fit the model, generate stimuli, and then as quickly as possible, come back to the same animal and then show back those images. And we had these Utah rays that were already implanted on the cortex. And these Utah rays, they have relatively consistent, reliable data over a period of like months. If you're lucky, maybe up to a year and so on. So that was kind of like a good insurance guarantee for us that we're not gonna lose all the units that we were recording from in this first session, because that's really important to make sure that we're recording from the same neurons roughly between the two sessions. And another thing that we found out was that the quicker they could close the loop, the higher, the more likely it was to actually succeed in control. Meaning that if we close the loop in a matter of like a day or two, we would have expected much better results rather than going for 2 weeks or so. Which was the initial experiments were actually kind of like that. They were slower because we were still experimenting, like doing this and then trying this multiple times, go back next week. But the latest monkeys that we tried, it's like closing the loop within a day.
Yeah, quite challenging. Dylan, you actually have your microphone on. Did you have a question? Nope, could be a chance. Curtis, what about you?
Yeah, I had another question based on what you just said. Is it conceivable some of the neurons drift over time in their preferred or in their supernormal stimulus, so to speak?
What do you mean by drift?
Well, so there were earlier studies, this is kind of way outside my area, from people in Japan. I want to say, no, sorry, I'm not sure I remember the name, where they showed that IT neurons that had complex pattern preferences, that preference could dramatically change depending on the context of a behavioral task. Now, I know you don't have a behavioral task, but these monkeys went back to their cage every day and you know, maybe were socializing with other monkeys or eating different foods or, you know, playing different with different toys or something, and they might come back the next day and the preferred stimulus might be different, or maybe not. I don't know.
I see. We did not try this exact experiment of saying whether the preferred stimuli with synthesis changed or not, but we did try two things. One was that we were always looking at the consistency of responses across sessions. So we had this set of stimuli called normalizer set, which was 25 images that we were showing every session. And then we were looking at the correlation of activity across sessions for those images. And we actually, in our analysis, we always only looked at those neurons that they had high reliability, high consistency across sessions. So even if there were such neurons that were drifting, as you're saying, we were choosing to discard them because it was very important for us to be looking at the same neuron from session to session. And some of these driftings could happen just because of like tiny movements in the Utah ray or like monkeys fighting in the cage and everything could go on and so on. So we're trying to like filter those things out. And the second thing that we did try around the same time was having monkeys doing the passive task versus doing kind of like a match the sample task, which is kind of like a more active version of this. And we did not notice any difference in the neural patterns of responses across the two tasks. They were very similar to each other. So the neuronal selectivity did not change across tasks, at least these two tasks.
Right, thank you.
Another question. I think we'll be wrapping up soon. One last question from anybody? Okie doke. Pouya, thank you so much.
Thank you for inviting.
Interesting work with some, very fascinating kinds of results coming out of it. So I look forward to hearing from you again in a few years' time and see where it's gone. Thank you so much.
Bye, everyone.
Bye-bye.